Hi there. My name is Xander, and I work for a company in London called Synthace. We're a startup synthetic biology company. We're actually one of the first UK companies that was a solely synthetic biology dedicated company. It was founded a few years ago by a bunch of scientists who really wanted to optimize the way that we do biology to apply the processes of engineering to improve the process of biological manufacturing, to improve the output of any sort of biological process. And the reason they wanted to do this is because we think that a lot of biology can be done in a wrong way, that there's a lot of scope to improve the output we have in our biological processes. You're familiar with Moore's law, but in research and development there's also a concept called Ehrum's law, which states that over the past 70 years we've invested a lot more money into the development of drugs, but the actual amount of drugs that we've obtained that are going through clinical trials that are viable has actually decreased. And one of the things that should be happening is this curve should be going the other way. We know a lot more about biological processes nowadays. The cost of DNA synthesis is a lot cheaper, but there's a lot of reasons why we are seeing this decline. And at Synthace, one of the reasons we think is the main cause of this is that a lot of biology is not reproducible. It's quite well publicized now that a lot of biological processes and methods are not reproducible. For example, only 25% of pharmaceutically published papers have their results replicated. Only 11% of cancer research papers have their published results replicated. Just this paper alone looked at a random sampling of 441 biomedical journal articles and found that only one study provided a fully detailed protocol. And isn't that just quite overwhelmingly bad? You know, science is about openness and peer review and being able to take people's methods and reproduce them, but actually it's not really the case. One of the reasons for this is that when we write biological methods, they can be quite ambiguous. You know, we record our methods in laboratory notebooks that are stored on shelves for years. We get data from these experiments that are poorly recorded. We don't then take that data and use it to feed and inform our experiments in the future. You know, and it's not just the case that it's just because this is written out in a funny way. It's actually part of, you know, the language that we're using to describe our protocols. You know, it can be quite abstract and ambiguous. What does, you know, shake mean? What does incubate at whatever mean? The human language itself has this abstraction that's quite hard to pin down. You know, I remember in my graduate research when I was trying to replicate a protocol from a previous postdoc, he'd said things like, you know, prepare the samples, pipette them into tubes, place them in the PCR machine, and set shaking at 800 RPM. And if that's not clear to everyone, the idea behind that is actually a PCR machine should never be shaken. They're not meant to shake. But you don't sort of get this from the protocol, because in their own mind when they were writing this protocol, that made sense to them. It was their protocol, but that protocol is not transferable. So this idea of transferability of protocols is really important, because there are a lot of things that could influence biological processes that may not be obvious to us. And these can be quite small details, such as, you know, shaking at 800 RPM instead of shaking at 200 RPM. We assume that it's going to be okay, but it might be a really important factor. To give an example of this, construct assembly, so the way that we splice together bits of DNA to test, it's kind of one of the fundamentals of synthetic biology. At Synthace, one of the methods we use is called Golden Gate, which is where we use a type 2S restriction enzyme that cuts away from its recognition site. This allows us to create modular types of assemblies where we can assemble lots of different pieces in a unidirectional way, and we can create lots of different constructs. Essentially, it's cloning, and cloning is awful. You know, people don't like to clone. Scientists don't like to clone. It's one of these things that you have to do. It's a necessary must to get to your data, to get to your experimental result. And it's something that scientists really don't want to be doing. And actually, cloning is quite difficult. It doesn't work a lot of the time. And why doesn't it work a lot of the time? one of the methods that we like to use is one that's been used in engineering for a long time. It's called design of experiments. It's where you identify a lot of the factors that might be important to your experiment. And you lay them all out and you plan an experiment where you're essentially testing all of these factors and measuring their interaction between each other, because actually we have this mindset that, oh, maybe the DNA concentration is a really important factor, but we don't think about the fact that the DNA concentration might be interacting with something else, like the temperature or the size of our DNA parts. So we conducted this experiment where we looked at 19 factors and we looked at construct assembly. And we realized that actually in over 50% of the runs that we were doing, we were getting colonies, and actually the majority of them had the wrong construct. So once we identify the factors that are important, we can then hone in on the ones that we think are essential, and we can refactor the experiment. So we're properly recording the experiment, we're taking the data out of it, and we're using that data to reinform a further iteration. So if we look at the conditions under which we knew that we were getting high levels of the correct construct, on a second iteration of the experiment, we were then able to attain, for example, 100% of runs where we were getting more than 40 colonies, and 99% of those colonies had the correct construct. So even though something is not working, we can improve it if we know how to look at these factors and how to identify and characterize these factors, and to understand that these factors can interact with each other. At Synthace, we've been developing a programming language called Antha, which is something that we're using to improve the way that we're doing these biological workflows. It's a high-level language, so it's interested in the concepts of the experiment you're doing. You don't have to worry about, for example, the concentration of your DNA. You have to worry about actually doing the assembly, doing the transformation, doing whatever experiment. So Antha is separated into two branches. We have the programming language, which we call AnthaLang, which essentially details the code of the experiment. So we call these elements. Each experiment or each experimental method has an element into which are encoded parameters that are important for that experiment, which are strongly associated with types. And you can define these types so that these types then associate with, for example, predefined parameters. It gets rid of the abstraction of having to write out things and, let's say, be ambiguous about what they might mean. So if you have a DNA part, it always inherits the qualities and the parameters of a DNA part. We also have an Antha OS operating system, which is, it takes away from the coding, and that you don't have to deal with code. You can execute your experiments through an operating system. You can create workflows, and you can initiate experiments, track data, form inventories. But the really useful thing about the code is this idea of it being reproducible, and that once you conduct an experiment using, for example, AnthaLang, that experiment is traceable. You can go back to that experiment that you did on the 22nd of January, and you can know exactly what parameters you used for that result. It takes away from this ambiguity of not knowing what went wrong with your experiment, but being able to pick out the things that did go right and being able to reproduce them. We also understand that automation is really essential for the future of synthetic biology for two reasons. One is high throughput. Machines are great at doing lots of things, lots of times, but also reproducibility. You can't trust a human to pipette out 5 microliters 5,000 times. There will be a great margin of error, but you can trust automation to do it. So what we're doing is we're developing drivers for lots of different bits of liquid handling platforms, and we're using these liquid handling platforms to facilitate our biological workflows. And it's not just using these machines as liquid handling to do one process, it's using these liquid handling machines to do whole experiments. So no longer would you just use it as a plate setup, you use it to do a construct assembly and an incubation and a transformation. Antha is something that can control lots of different bits of kit that are useful to your experiment, like liquid handling, like an incubator that you might use to recover cells on at 37 degrees shaking. An example of this is we recently did a project for Merck Bioscience in America, where we used quality by design. You know, this idea of making sure your experiment is really well designed before you before you start it. So they wanted to look at lots of different vector constructions. So we designed these vector constructions, and from the moment we got the DNA in, we used Antha to execute the experiment, to track the samples, to execute the automation, to do the transformation. And from receiving the DNA samples to having verified constructs, we did all this in 3 days. So having that reproducibility where we've already optimized these procedures to a point where we can get in constructs that are novel to us, but we're confident about the way we're doing them, that we can get to these results in a span of days. In engineering and technology, it's quite common to see technical data sheets. So this is just an example I've pulled from the internet on reflex transistors, where if you buy a component, it will have been tested to such a way that you can identify the conditions under which it will work really well or under which it will fail. And we think we can do this with biological methods. So for example, construct assembly. We've done extensive robustness studies on DNA construct assembly. And this is just an example of an output of this, where we've changed lots of different conditions and looked at all the interactions between these common conditions that you might see and that you might encounter when you're doing a DNA construct assembly procedure, and being able to pinpoint exactly where it would go wrong and where it would go really well. Consequently, we've been able to go from a really long process of construct assembly, where you might have your parts in, it takes a long time to design, it takes a long time to do the actual procedure, to a much shorter procedure where instead of worrying about these individual parts where you're having to linearize your vector or mix all your parts together, you can use something like Antha to automate your experiment. What's also quite important about this is the transferability. I mentioned earlier on that it can be difficult to go back to experiments and know, for example, exactly what you did, to have it all laid out. But using a coding language like Antha means that, for example, someone in America can replicate your experiment and you can know exactly what they're doing and exactly when they're doing it. We recently did a contract for a company called Dow AgroScience in America, and they wanted an automated procedure. And the fact that we're using the Antha language to automate that procedure means that we can know exactly what they're doing. We can identify the things that they're trying to analyze. We can make improvements, and we can send them to them in an electronic format, and they can execute them. It also means that we can think of these biological processes as workflows instead of individual experiments. So for example, if you had an Antha element that would make a master mix, you could hook it into a colony PCR setup. You could then do the PCR reaction and use an automated component to load the gel. So this whole workflow would be your colony PCR workflow. You could do the same with the assembly of constructs and transformation, and you could do the same with culture inoculation and measurement. So I titled the talk Developing Tech Bio, and when we started the company, the company was started as a synthetic biology company, and we kind of crossed the barrier between synthetic biology and biotech. But when we started developing Antha as a programming language, our CEO Tim came into the office one day and said, he was really passionate and really excited. He said, guys, we're no longer a biotech company, we're a tech company. And we kind of understood that because we kind of bought into the idea of Antha and what it could do and the importance of having these codifiable elements. But we've since sort of moved to this idea that we're more of a tech bio company that uses the tech concepts to improve biology. And I know I can rave about automation, I can rave about coding and how it's improving our biological workflows, but actually, in the current situation, it's really a tool for scientists to help their biological workflows. I can take out my phone and I can open up a travel app, and it will tell me the cheapest way to get from A to B and the quickest way to get from A to B, but it won't really tell me where to go. So the idea of automation in biology at the minute is really a tool for scientists to facilitate their workflows, to allow them to have critical thinking, to analyze their data, and that's really a fundamental importance to improving biological research. I've got a few minutes, so I just thought I would give an overview of, let's say, how we would interact with Antha, how we would do it at Synthace, how other people are doing it in places like Merck and Dow. We have an operating system. We have two types of operating system, one of which is this sort of card protocol, where we have already predefined workflows. So here, for example, we have a Type 2S construct assembly workflow. So if you were to click that button on the right-hand side, it would then lead you to being able to upload a file, a design file. It will then lead you to the inventory of things that you would need for that experiment. It will check with your inventory in your lab to make sure that you have it. It will then tell you all the things you need to do to prep the plates for it. It will then execute the experiment for you on a liquid handling platform. If incubators are needed, it will control the incubators. Once it's done, it can then take in the data, the plate outputs from that can then be used in other experiments. But we don't like this idea of just having fixed protocols. We like people to adapt their protocols to their own needs. So we also have a workflow editor. And this workflow editor basically consists of different Antha elements. And each Antha element is its own little experiment. So what you can see here is I just took a screenshot of where I linked together. The middle one is a construct assembly. On the left-hand side, we have a master mix maker, where it will make your master mix for you. We also have a protocol that will help you design your combinatorial assembly. So essentially, you're inputting your master mix, the actual liquid components for your experiment, and the design files, the sequence files for your experiment. And at the end, on the right-hand side, we have a transformation element, so it will do your transformation for you. We can also use visual graphics to, let's say, troubleshoot the experiment to make sure that it's doing everything we want it to do. So if we simulate this whole experiment, we can track all the liquid handling components to make sure that they're going to the right place. And if there are errors in the process, if something does not go quite how we expect, it's traceable. We can go back and we can identify exactly where it went wrong. And this is an example of a deck layout. So Antha will dictate to you exactly where plates have to go, and this is on a Gilson Pipette Max robot. It will say what rises it has to go on. It will say that in position 6, for example, you need to put it on a cooler. On position 1, you need to put it on an incubator. It can then control the incubator for you. It tells you what tips you need and where everything needs to go. So that was just a quick demo of the Antha operating system. But the Antha language itself is an open source biological language that you can access yourself. You can find out our GitHub address is on anthalang.org, and you can find out more about the company at their website, synthace.com. So thanks very much for listening, and I'm happy to take any questions.
Thank you, Xander. That's a question here from the boss in front. A moment.
Hi, thank you so much for the talk. Really enjoyed it. So I have two questions. The first is it possible that you can create an interface with existing lab equipment, like, for example, your incubator, shaking incubator, and all this? And the second question is that, so, for example, myself, I'm not adept at coding at all, unfortunately. So do you plan to create any user interface to make this more intuitive?
Yeah, so the first question, so we're developing drivers to control various bits of kit. So at the minute we can control things like an on-deck shaker incubator. So it absolutely can control stuff like that, and it can integrate the control with, for example, the liquid handling robot. So you can control those independently for your experiment. And the second question is, we already have this sort of interface. So actually the interface I was demonstrating is our operating system. So a user who isn't experienced in coding could completely belie any need to go into the code. They can use either these card protocols that we're already developing, or they can hook their own protocols together via the workflow editor. And those can be bespoke protocols. You know, you can do the transformation that I demonstrated, or you can do, you know, preparing a PCR plate and then running a gel just by hooking it together. Conversely, you know, I mean, I think programming is a really good skill. Until over a year ago, I didn't have any programming knowledge, and so I had to learn it through this job position. And I think it's a fantastic skill to have. So I would kind of, you know, like push on anyone who is interested in it to go forth and do it. But if you are to go with Antha, and if you were to look at the open source language you have, then you would need a certain programming knowledge. But you know, our user interface is something that you could use and never have to deal with coding.
Excellent. Thank you.
So I suppose you may have actually just answered this question, but what is the next step for the company? You clearly have something that's working, and
yeah, so the company is, so we've currently deployed Antha into Merck Bioscience and Dow AgroScience in America. So currently we're looking at potential clients, other big pharmaceutical companies where we can deploy the Antha OS user system. We're also developing further protocols that can be put into this card system. So we have this workflow editor where we have these protocols that are defined and connected. But people are quite interested in this card system where instead of having to worry about putting things together, they can just click, make me this construct, and it will do this for you, this high-level concept. So we're looking more into that, and a lot more protocol engineering, a lot of developing the interface of the software, developing like the user interaction with the software. We've already gone through quite a few changes of that in recent months. In terms of traceability of, let's say, samples, traceability of plates and storage of consumables as well.
Just in terms of, I mean, you mentioned the big companies there that you're sort of working with. On what level are they sort of engaging in this now? In terms of, is it like right across the department with them, or are they sort of developing it at a small level in-house to which they'll sort of expand, or?
At the meantime, it's particular like groups within American Dow AgroScience who've decided that this is something that they want to use to automate, for example, their liquid handling robots to automate their biological processes. So currently it's at, let's say, particular research groups within the companies that are interested in what we're doing, who have contracted us because they want Antha, because they want, let's say, bespoke automated protocols, and they're now using Antha to control their biological processes and to implement those protocols.
And of course, the embarrassing question of money. Is this partly the reason you're dealing with bigger companies now? Is it sort of a substantial cost?
Yes.
Yeah.
We've kind of went through different pricing structures over the past year because we wanted it to be accessible to, let's say, lots of different sizes of companies. But actually, like, you know, there's a lot of costs associated with it.
Any other questions? Yeah. Sorry.
Yeah, thanks for your talk, Xander. Bearing in mind that 90% of all candidate drugs fail before they reach the lab, are you getting more and more involved in personalised and precision medicine?
We haven't dealt with any, let's say, medical or precision medical companies.
So just following on from his question of future developments, is it something you're considering working in that area or moving into that area?
Not currently. The current, let's say, clients we're dealing with are dealing with more like biomanufacturing, increasing like process output of different components that go into different manufacturing processes, but currently not medical.
And of course, you mentioned the rather sort of strange way of recording lab data. Are you heavily involved in ELN as well, making your systems? Are they integrated with that?
It's ELN?
ELN.
What's ELN?
Electronic laboratory.
Notebook. Is that what you mean? Electronic?
Yes. Yeah.
I'm not familiar with what that is, electronic laboratory.
Electronic laboratory notebooks, ELN, sort of software we can automatically record your data.
Yes, yeah.
So those are different names, but that's what I know.
Yeah, so we're currently developing our own in-house method of, let's say, recording biological protocols of
It's for the
recording.
sake of reproducibility, because you mentioned that.
Yeah, exactly. So we're currently developing our in-house method of recording, let's say, how experiments are done, but also because, for example, experiments that can be executed via Antha are very traceable. So to execute an experiment, you need to already predefine, let's say, the parameters for that experiment. So essentially, once you execute it, you have the code, you have the parameters for that experiment, so you can already trace back to it, and that kind of functions as your method for that experiment, really.
Yeah, because you are talking about reproducibility and trying to standardize certain sort of lab recording techniques, are you hoping that Antha will sort of be adopted sort of on a wide spectrum, like academically, industrially, sort of?
Yeah, absolutely. Let's say the feedback and like positive reception we've received from, let's say, industrial partners is quite substantial. They see the need for this. They see the need for something like Antha that can control different bits of kit, that can automate their processes, that can get rid of any reproducibility issues. We also want it to be adopted, let's say, academically. We want people who are passionate about this idea to get involved in it also. So we would definitely hope for it to be become more widespread.
Okay, I'll start you, Sylvain.
Sylvain from Genopole in France. So really nice presentation. We've already seen that before, the open language. Just to say, it was really not a question, just that we've developed that. We've developed this part of the user interface much more hand-based for the users to automate workflows and stuff, and we'd love to partner. We have the ELN part and stuff, so I mean, just to be sure we have lunch together and that we meet.
Oh yeah, absolutely. That sounds good. Let's chat.
Okay, is that everybody? Everybody's sorted for lunch, so.
Okay, thanks very much. Cheers.